I am Dr. Khokon Kumar Dutta. My Christian name is Dr. David Moon. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is MicroRNA-34c is a critical player in restoration of impaired memory formation in human brain. Khokon Kumar Dutta et al. For the first time succeeded to validate the existence and expression of precursor forms of MicroRNA-34c in the lung and testes of rats. The group also succeeded to verify the involvement of MicroRNA-34c in rat renal tubular cell carcinoma, mouse embryonic stem cells, and in mouse embryonic fibroblast cells. Internationally, this was the very first example of experimental validation of expression of MicroRNA-34c in any living organism or in any in vitro system. We published the findings in 2007 in the Journal of Cancer Science entitled Association of MicroRNA-34a overexpression with proliferation is cell type dependent. Subsequently, other scientists succeeded to discover the expression of MicroRNA-34c in different other cell types and organs. They also succeeded to explore the critical functions of MicroRNA-34c in different physiological and pathological processes. Athanasios Zovoilis et al. Published an article in 2011 in the Journal of the European Journal of Molecular Biology Organization, EMBO, entitled MicroRNA-34c is a novel target to treat dementias. Dementia is known as the loss of the ability of memory formation in the brain, language, problem solving, and the loss of the other thinking abilities. Dementias are a frequent part of different neurodegenerative diseases like Alzheimer's disease. The group found elevated presence of MicroRNA-34c in the hippocampus area of the brain of human Alzheimer's patients and in the hippocampus of mice without disease in an old age. The level of MicroRNA-34c was correlated with the degrees of cognitive impairments or learning disabilities. The memory function was significantly improved in Alzheimer's patient or old age brain only in 36 hours after 3 consecutive injections of the inhibitor of the seed of MicroRNA-34c. The group reported that SHRT1 is the target of MicroRNA-34c in the hippocampus of the brain. SHRT1 is known to have involvement with the memory function of the brain. At least a part of the mechanisms by which MicroRNA-34c regulates the memory function of the brain is by targeting SHRT1. The group also reported the connection between MicroRNA-34c and contextual fear conditioning. MicroRNA-34c level was transiently elevated just after 3 hours of exposing mice with contextual fear conditioning, which is followed by the wave of expression of the target protein coding genes. Roberto Fiore and Gerhard published an article in 2011 in the Journal of European Journal of Molecular Biology Organization, EMBO, entitled "Releasing a Tiny Molecular Break May Improve Memory." They reflected on MicroRNA-34c as the break in the memory formation process. The Daily Express reported on MicroRNA-34c on September 24, 2011, with the title "Secret of Restoring Dementia Victims' Memory." Shephali Bhatnagar et al. Published an article in February 2014 entitled "Increased MicroRNA-34c Abundance in Alzheimer's Disease Circulating Plasma." Yu-Chia Kao et al. Published an article in August 2018 in the International Journal of Molecular Sciences entitled "MicroRNA-34c Overexpression Causes Dendritic Loss and Memory Decline." The group found that high level presence of MicroRNA-34c in the hippocampal neurons negatively regulated the dendritic length and spine density. They also made elevated presence of MicroRNA-34c in the hippocampal neurons using transfixion of MicroRNA-34c and found shorter dendrites and on average and spines than those not transfixed with MicroRNA-34c control group. To mention here that dendrites and synapses are key sites for signal transduction and fundamental structures for memory formation and storage. Elizabeth M McNeill et al. Published an article in February 2020 in the Journal of Nature Communications entitled "The Conserved MicroRNA-34 Regulates Synaptogenesis by Coordination of Distinct Mechanisms in Pre-synaptic and Post-synaptic Cells." The group found that MID34 limits the expression of the junctional receptor CNT-NAP Nrx-IV to regulate the presynaptic features in motor neurons, and MID34 limits synapse growth by regulating the F-actin-associated isoforms of adenosine HTS in postsynaptic muscle cells. Null mutations in MID34 display combined defects characteristics of Nrx-IV and HTS-M isoform deregulation on the two sides of the synapse. Yiqing Huang et al. Published an article in January 2018 in the Journal of Molecular Medicine Reports entitled "Role of MicroRNA-34c in the Cognitive Function of Epileptic Rats Induced by Pentylene Tetrazool." The group induced experimental epilepsy by injecting pentylene tetrazool and then treated with antisense or antagonist of MicroRNA-34c. To mention here that increased deficit of learning and memory function is a period of clinical epilepsy, which is associated with seizures or periods of unusual behavior, sensations, and sometimes loss of awareness. The study also found that long-term potentiation LTP is one of the principal pathways of MicroRNA-34c in memory formation. Finally, I would like to wish good health, prosperous and joyful days for you everybody. Thank you.